Australian University Department. He brags of a Nobel laureate, an Australian prize, a Federation prize, 13 professors, and at one stage, 5 National Master Program Prize. And it's always been bandied around that in that department, Frank Carboni got the best students. If you listen to Scott, you understand why that rings around. Scott did his PhD with Frank, finished up at the end of 2003, publishing a James paper and a few other papers, including a JI paper, which I think is really extremely notable, but no one cites it. Then went on to wrap out into the postdoc at Emory. Did okay, didn't he? Got science, PNAS, JAGS Med, JAG Clinical Investigation. In fact, he did so well that when he wanted to learn 2-photon imaging, Racker was quite happy to pay for him to go and use a non-germane slate at the NIH for 1.5 years. And, if you knew Racker, and if you fully funded that, you would understand why he really thought of Scott. Scott has, amongst other qualities, I think the 3 signature qualities that Scott has is that he's expeditious, he's indefatigable, and perhaps most uniquely, he's actually quite a silky writer. Anyway, so Scott's going to talk about recent work he's done. He's been back for a year and a half or so. 2. In that time, I've got an age paper out. He's going to talk about that today. Scott.
Thank you, Andrew, for that lovely introduction. So I think I'm starting on a high note then, it'll be downhill from here. So I'm just sort of going to give an overview of the work that I've been doing, and as well as some of the other people in the whole Heath Carboni empire that's all sort of been contributing to the general project that we've been working on, looking at the dynamics of T-cell responses in the skin. Thanks. So just to give you a bit of background, probably a lot of people here are very familiar with the model, but in case you're not, we use a cutaneous herpes simplex virus infection model by scarifying the flank of a mouse that's been shaved, and then the scarification is done with a Dremel power tool, which always seems to tickle people. The virus, of course, infects locally, mostly the keratinocytes and the epidermis. It then enters the sensory ganglion and is transported to the dorsal root ganglion, where it undergoes further replication. And then it spreads throughout the innervated dermatome by coming out and essentially, initially at the site of replication, what we call the primary site, it forms a lesion, but then it goes into the nerves and comes back out and forms a band-type lesion throughout the innervated region of the skin. And what's really powerful about the model is there's quite a few tools we can use, but the 2 main ones that we make a lot of use of were made initially quite a few years ago by Frank Carboni, and I was part of that. It was a GBT1 mouse, which is a TCR transgenic class 1 restricted mouse specific for the glycoprotein B from herpes simplex. And then more recently, in conjunction with the Andrew Brooks lab, a CD4 transgenic class 2 restricted responding to an epitope from GD. And by using these 2 TCR transgenic mouse lines, we've been able to do a lot of quite sophisticated in vivo work, which is what I'm going to go through now. And so this is work from Liv Eidsmo, who was in the lab, just to give you an idea of the kinetics of the T-cell responses that we've been able to track using these TCR transgenic mice. So if you look in the draining lymph nodes, we initially see expansion of the CD8 T cells, GBT1 cells in this case. First in the brachial lymph nodes, it's the primary draining lymph node. And then following that initial expansion, you get subsequent appearance of cells that move from the brachial lymph node via the lymphatics and start appearing within the secondary draining lymph node, which is the axillary lymph node. You then start to see expansion of cell numbers in both of those compartments, after which the cells then get into the circulation, and you find them in the spleen at around day 5 to 6 after infection, where they undergo a massive number of proliferation events, expanding the numbers of CD8 T cells that you get. And then following that, the cells, of course, get into the skin. This is a graph from Thomas Gebhardt, just showing you that somewhere between day 5 and 6, after they're released from the lymph nodes and after they divide in the spleen, they start to appear in the skin, and that's where they do their job. And we know that the appearance of T cells in the skin, somewhere around day 5 or so, is the initiation of the clearance of the virus. And then within about 7 to 8 days, the virus is essentially cleared from the skin. Replicating virus is also cleared from the dorsal root ganglia, although of course the virus then goes latent in that site. Now, with the arrival of the CD4 TCR transgenic mice, the GDT2s, we could also start to track the CD4 T cell response. And this is some work done by Paul Whitney, who made the mice. And you can see essentially that the expansion of the CD4 T cells initially occurs in the brachial lymph node. We really first start to see cells appearing around day 5. There's a subsequent appearance of cells in the axillary lymph node and then the spleen, so that the kinetics of the response is essentially the same as for the CD8 T cells. And then after the large numbers of cells get into the circulation, somewhere around day 5, we start to see them appearing in the skin, potentially slightly earlier than the CD8 T cells, which is interesting. But again, it's around that time point, 5 to 6, when the cells start accumulating in the skin, that we know that the virus begins to be cleared. Now, all of this is well and good, but what I was really interested in, initially at least, was what is happening in the skin once the cells get there. And this is just to give you a sort of a very quick reminder of the structure of the skin. The outer layer, of course, the epidermis, it consists of a number of different sublayers. The outermost layer, the stratum corneum, is essentially the dead skin cells. And then you have basically 2 layers in mice, the stratum granulosum and the stratum spinosum. The stratum spinosum is the basal layer where all the main keratinocytes are and your immune cells, which I'll show you later. This epidermis, of course, is then separating the dermis. It's separated by a basement membrane, and hair follicles penetrate from the lower dermis all the way through to the epidermis, but they're also separated from the dermis by a basement membrane, essentially making the hair follicle structure more contiguous with the epidermis than it is with the dermis. And of course, we know that throughout the dermis, snakes, blood capillaries and lymphatic vessels and various other things that provide its function. This is a cartoon from human skin, but many of the structures are the same in mouse skin. And we know that from doing just various immunofluorescence stains on sections of mouse skin, it would allow us to detect the various structures in the skin. Namely, we can see the basement membrane that separates the epidermis from the dermis. We can detect the blood vessels and the lymphatic vessels, and this big blue structure is a hair follicle, which is dense for the DAPI-positive cells, and we can see networks of fibroblasts, the dermal fibroblasts throughout the dermis. Now, to analyze the T cell responses in the skin, of course, we wanted to be able to see the T cells by microscopy, and to do that we crossed the GBT1 and GDT2 mice to mice expressing either GFP or the red fluorescent protein DS-red ubiquitously. And so what we essentially do for most of the experiments that I'll show you is to transfer these TCR transgenic T cells into mice, followed by infection, and then at various times afterwards to image the skin by either confocal or 2-photon microscopy. And what we saw when we did confocal microscopy on the skin, this is 8 days after infection, so this is actually the point when the virus is essentially completely cleared. But what you can see is that both the CD8 and the CD4 T cells have infiltrated all the regions of the skin. There's large numbers throughout the dermis. There's quite a few scattered throughout the epidermal layer, which is labeled with keratin 14 in this case. And this is essentially the crusty lesion that forms over the skin, and you get large clusters of both CD4 and CD8 T cells in this site. no doubt doing their job clearing the virus. But of course, what my primary interest was is not just to look in this static situation, is to really understand how these T cells are actually behaving in the skin following infection. And so, as I said, we use 2-photon microscopy for that, and I don't want to get into too much technical detail, but I thought I'd just give a bit of an overview if you're not familiar with the general principle behind it. Typical single photon excitation of the type that you would do for most imaging, such as confocal imaging, you use a laser or a light source that will provide enough energy to excite the fluorescent molecule, and then of course it emits its fluorescence in whatever color it is. 2-photon simply works on the principle that you use a much higher wavelength light, and the only way to excite the fluorescent molecule is to have 2 photons of light simultaneously hitting your fluorescent sample to combine together to form enough energy to excite it, and then you get the same fluorescence. And the reason that is, the hardware behind that is very similar in that if you consider a typical confocal microscope, you shine your laser on your sample, you get fluorescence. This is collected after first passing through a pinhole, which essentially discards out of focus light. So you get a slice image, and the detector only picks up that confocal slice. Whereas with 2-photon, your laser excites your sample, and then you have an external detector that collects all of the light that is coming from your sample. But the reason why that's powerful is that in contrast to single photon excitation, where the laser essentially shines all the way through your sample and is exciting the sample all the way through as it goes, and much brighter at the front of the sample, 2-photon will only excite fluorescent molecules at the point of laser focus. So it in itself is essentially confocal in that it's only exciting at the point of focus. And what's particularly good about that is by using a long wavelength light, we get much better tissue penetration, less light scattering, and much less photo damage in the tissue. And that, of course, allows us to look in live tissues for longer periods and see quite deep into tissues, whereas with typical single photon excitation, we generally can't see very deep. So it's a great tool to allow us to look into tissues of live animals, and we started by doing skin imaging, as you would have guessed. We have a Zeiss 2-photon system and a custom-built incubation chamber, which is kept to 35 degrees. So essentially what we do is we keep an anaesthetized mouse in the chamber, and we can image it for a minimum of 4 hours, up to 8 hours if we like, when the mouse is quite happy and quite healthy. And what we do for the skin imaging is we do 2 small incisions on the flank in order to raise the skin slightly and stabilize it with this metal plate to prevent movement from breathing. And then we keep the mouse anaesthetized, and we can image it for hours. So when we first started to look at the T cells in the skin, we were really impressed by how dynamically both the CD4 and the CD8 T cells were moving around in the skin. The other thing I should note is that another good point about 2-photon microscopy is that it non-specifically induces fluorescence in collagen-rich structures, and the dermis, of course, is highly enriched for collagen. So we can see the dermis in all of the skin imaging movies because it comes up blue, and this is known as second harmonic generation. And what you can see here is that there's both CD4 and CD8 T cells zipping around the skin. We can see some moving very quickly, others clustering around hair follicles, and yet others moving in quite interesting slow patterns. And so we were quite impressed by how dynamic the movement of these T cells were in the skin. What's interesting is if you take a movie, this is looking from the top obviously, if you flip it on its side, you can get a better idea of where the T cells are in the different layers of the skin. The epidermis, of course, being low for collagen is coming up black here, and the dermis coming up blue, so we can tell where we are. And this red thing here is this hair shining through here. And we can see that there's T cells present in the epidermis, many in the dermis, and also, interestingly, you get T cells infiltrating into the epithelial layer of the hair follicles, and they move around there as well. So they're really infiltrating the entire skin. And what we found interesting was that the movement of the cells really appeared to differ depending on where they were. If we focus on cells that are in the dermis, they tend to be moving much faster, whereas cells in the epidermis appeared to be considerably slower. This is tracking CD4 T cells, The dermal T cells you can see with the purple tracks are moving very quickly, whereas the epidermal cells move considerably slower. And that was for CD4 T cells, and the same is true for CD8 T cells. Those in the dermis are moving quite quickly, zipping through, whereas those in the epidermis move with a much more pronounced slow movement. And tracking analysis confirmed this, that whether it be a CD4 or CD8 T cell, its presence in the dermis allows it to move quite fast in a range of around 5 to 10 micron per minute, and it's at least half that in the epidermis. So there's a differential migration depending on what part of the tissue they're in. And we were interested, and we still are interested, in trying to understand how the T cells get from the dermis to the epidermis, because of course it's, at least in a healthy state, it's separated by a very pronounced barrier, that being the basement membrane. Obviously, during the infection and the inflammatory reaction that occurs, it's possible that that membrane breaks down. But we still had trouble actually tracking cells making the transition from the dermis to the epidermis. And we found a couple of examples, but very, very rare, where you can see a cell coming along in the dermis, and then it pops up into the epidermis. And as soon as it does that, it essentially slows down quite dramatically. And if you track the speed of that cell in the dermis versus when it's popped into the epidermis, the speed difference is exactly as we saw when we tracked the different compartments before. But it's interesting that despite seeing very large numbers of cells in both compartments, we hardly ever see the transition between the compartments. So my interest for a long time has been in T cell memory, and we know from work done by Thomas Gebhardt that there is a population of CD8 T cells that reside in the skin. So what we were interested in doing is seeing what they look like and how they behave in the skin. And I don't need to really give you a background on memory, not to this audience at least. But we do know that the migration of effector and memory T cells is really relies upon the type of molecules that they express. If they express the right chemokine receptors and adhesion molecules, they can then get into the skin. And traditionally it's thought that CCR4 and CCR10 are critical to get cells into the skin, whereas things like CCR9 and so forth are critical for the gut. But what's really not known is why some cells remain in certain tissues and why some cells continue to migrate around the body. Now, of course, in terms of memory T cell subsets, it's been known for a long time that there's at least two memory T cell subsets, namely the effector memory population, which is largely replication incompetent and is thought to transition over time, at least by some people's models, into a population of central memory T cells, which attain the ability to homeostatically proliferate and divide much better upon antigen restimulation. And these two subtypes are generally defined on the basis of CD62L and CCR7 expression. Namely, effector memories are low for 62L and low for CCR7, and vice versa on central memory T cells. But of course, from work done by Thomas Gebhardt and now others looking in different tissues, we know that there are populations of tissue-resident memory T cells that appear to be retained within tissues and are long-lived in that site and potentially don't follow this same pathway of differentiation. We know that central memory T cells tend to recirculate through lymphoid tissues and the blood, and there's very few in non-lymphoid tissues, whereas effector memory T cells tend to be able to recirculate through non-lymphoid tissues as well. Whereas the resident memory T cells appear to be just simply resident within certain tissues. And as I said, in the skin, but also in the gut, the brain, and the lung, and potentially who knows where else. Anywhere that has an epithelial layer might potentially have a population of resident memory T cells. So this is also initial work by Thomas Gebhardt, just to show that the tissue-resident population looks a lot like effector memory cells in a CD44 high, 62L, CCR7 negative, but it's also expressed CD69 and CD103, as well as the ligands that potentially initially enabled these cells to get into the skin. And the numbers didn't come up, but this is day 100. You see long-term residence of the T cells in the skin. at the site of prior infection. So when we started to image the tissue-resonant memory CD8 T cells in the skin, we were really quite surprised at how they're moving. You can see there's quite a range of shapes, but in general they're remarkably dendritic in their shape and their movement, and they're generally moving very slow, and as they move they tend to project out these really fine dendritic projections as they move through the epidermal layer of the skin. And at this time point you don't find any memory CD8 T cells in the dermis, although they do also exist. You can sort of see them here crawling along the epithelial layer of the hair follicles, which is, as I said before, contiguous with the epidermis. We were quite amazed at the shapes these T cells form and how they move through the skin, which is actually quite dynamic considering the fact that they retain there for such long periods. What was also interesting to note is the comparison between these CD8 conventional resonant memory T cells and the other population of T cells that's found in skin, at least in mice, the gamma delta T cells or the DETSI. They also form quite an interesting dendritic shape, but it's quite different from the range of shapes that we see that TRM form. And these have been shown to barely move in the skin in the steady state. They somewhere in the range of less than 1 micron per minute, whereas the tissue-resonant memory T cells are considerably more dynamic. And what's really interesting about the movement of these TRM in the skin is that they are restricted in a sort of a lateral fashion. They can put their projections out in all directions, except that we never see upward or downward projections forming, suggesting that they're essentially trapped within that first layer of the epidermis. The blue, of course, here is the dermis, and there would be a basement membrane running along here, separating this cell from its position in the epidermis to that in the dermis. And if we just apply a dye to the skin, CMTPX in this case, it labels the stratum corneum and stratum granulosum, and that defined the location of TRM in the skin to the stratum spinosum, or the basal layer of the epidermis, sitting right on top of the dermis and the basement membrane. And so, just to sort of reiterate the fact that these cells really do stay within the skin in this site, this was, I think, a really, really nice experiment that Thomas Gebhardt did, which I don't know if it was fortuitous or not, but he essentially used male T cells, put them into female mice, and you get expansion of both CD4 and CD8 T cells, and you get clearance of the virus, but then you get anti-male transplant rejection kicking in. And systemically, all of your T cells are rejected, so that in the spleen you no longer find either of these two populations at memory. But what you do find is the population of resident memory CD8 T cells in the skin, but you no longer find the CD4 T cells either, suggesting that the CD8s alone are sequestered from the circulation. They're not available to be rejected by circulating cells or circulating antibody, suggesting that they're really in a special location. And what I find particularly interesting as we're doing the tracking analysis of the TRM is that although their velocity is slowing down from a transition from the acute phase at around day 10 to a memory time point, the speed during memory is very consistent over a long period and very slow, obviously. But despite this slow movement, they are able to move a certain amount as they sort of project themselves through the tight networks of keratinocytes. This is an overlaid image of a number of TRM. It's taken over an hour-long period, and each image is taken every minute, and then you overlay the images and you get essentially a map of how much of the epidermal area the T cells can scan, or how much they're physically covering in that hour-long period. And we get a rough estimate of around 300 micron squared per hour that the T cells can actually scan, or area that they can cover as they move through the skin. And this suggests that if you extrapolate out to days, weeks, months, that their ability to move around, albeit slowly in the skin, allows them to essentially survey their environment for the reappearance of virus or their antigen at least. But what I find quite confusing in many ways is the fact that these TRM really do preferentially remain at the site of prior infection. We know, as I said, that they move, and if you do a displacement analysis on the T cells at various times at memory, on average they can cover around 200 micron per hour, suggesting that over a longer period they should be able to move quite a distance away from their initial starting site. But if we do mapping analysis and look at the proportion of T cells at the lesion site or a few millimeters away, either early after infection or late after infection, it demonstrates that the majority of the T cells remain right there at the site of initial infection, despite their ability to move around in the skin. They don't just diffuse randomly outwards and just get distributed all the way along the flank, suggesting that something may be keeping them there. And a good hint at that, at what is keeping them there, came from work by Azad and Nick in the lab when they were looking at DETSI, the gamma delta T cell populations in the skin. And this is an example at memory, and you can see the gamma delta T cells are stained in red, the TRM is stained in green, and they're essentially not localizing in the same areas, suggesting potentially that the DETSI are physically restraining the TRM into the location where they've seeded or the lesion site, preventing them from just randomly diffusing away. And this also suggests, interestingly, that there might be a competition for a local niche that restricts the numbers of T cells you get in the skin. And therefore, the DETSIs aren't able to come and repopulate this area, and the TRM aren't able to come and repopulate that area. There's a couple of other interesting things about the formation of TRM in that they express really high levels of the integrin CD103, which potentially suggested it was required either for their formation or their survival. And also there's suggestions that CD103 is key for the movement and the shape of cells within tissues. But when we looked at CD103 knockout versus wild type TRM, they both form in fairly similar numbers. They're in the epidermis, they form these dendritic shapes, and they move with basically the same velocity, suggesting that, at least in terms of them getting in and their ability to move around within the skin, CD103 is not a key molecule. And lastly, the other major population, of course, that's present within the epidermis is Langerhans cells. And now these dendritic cells form a really tight network, and when we image the Langerhans cells in green here and the TRM in red, you can see that there's quite a lot of interaction going on, and we're starting to quantitate this, but it appears that more of the time the T cells are in contact with at least part of a Langerhans cell than they're not. And that's probably got a lot to do with the really tight layer that's there with the keratinocytes not allowing much room for other cells to move in between. But it suggests that there might have been a lot of interaction or crosstalk between the two populations, and we thought that potentially the Langerhans cells might be a signal that allows TRM to either survive or move. But that's not the case, because when we used the Langerhans DTR mice, treated mice with diphtheria toxin for 2 weeks to deplete the Langerhans cells for a 2-week period, and then measure the numbers of TRM in the spleen or the skin, we found that essentially there's no difference between the treated or the untreated mice, suggesting that in the absence of Langerhans cells, the TRM are just as happy and just as able to get in and survive. And we also can show that when there are no Langerhans cells, the TRM still move around at a similar rate and have a similar shape. So at the moment we're not really entirely sure what drives the TRM movement. But one thing we do know is that the formation of these TRM in the skin is not antigen dependent. It's probably more likely to be inflammation dependent, because if we put in effector T cells either of an antigen-specific phenotype, the GPT-1s, or non-antigen specific using OT1s, which respond to avalbumin, put them into mice infected with HSV, both of them get into the skin and both of them form long-lived populations of resident memory T cells, which move at a similar velocity and have these similar dynamic dendritic shape and projections and so forth. So they both get there and they both form long-lived memory, but these guys didn't need to see antigen at any point in order to do that. Whoops. So that's all focused on the CD8 T cells that reside in the skin, and at the moment we're also very interested in looking at other factors that are driving their movement and their survival, such as chemokines. But the other thing that we were obviously really keen to study was the CD4 T cells, and we know that they get in the acute phase, but the question at that point was, do they also reside in the skin, or do they happen to do something different? And of course they happen to do something different, that's why we found it so interesting. This is just confocal analysis, and you can see that the CD8 T cells, of course, can be found in considerable numbers in the epidermis and also in hair follicle epithelial layers. But the CD4 T cells were really quite dramatically only found within the dermis of the skin after the clearance of the virus. This is day 30 as an example. So the localisation of the two memory T cell populations appeared to be really quite dynamically different. And they really were dynamically different when we did 2 photon and live imaging of the skin. This is 50 days after infection. The CD4s are in green and the CD8s are in red. As before, the CD8s are in the epidermis, they're dendritic, they're moving slowly. And the CD8s, the CD4s are in the dermis and they're really zipping around quite dynamically. And in these situations, we hardly ever see any CD4s in the epidermis. And we hardly ever see any CD8s in the dermis. And there's really a quite dramatic migration pattern that differs between the two populations of memory cells. This differential migration really tracks back to the physical location of the T cells, as I showed you at the effector phase, namely that the dermal CD4 T cells move really quite quickly as they move through the less densely packed dermis, whereas the epidermal CD8 T cells move obviously considerably slower as they move through the epidermis, and this is shown by tracking analysis. So again, it's really the location of the cells that's defining their movement, but the difference in their movement is really quite dramatic. And the interesting thing about that is that the CD4 T cells then were able to actually get out of the skin, and this was shown just by essentially taking a piece of skin, putting it into culture overnight. And you find a lot of the CD4 T cells are able to migrate out of that piece of skin into the dish, whereas the CD8 T cells are essentially trapped in the skin or sequestered, as I showed you before, suggesting that by being in the dermis, the T cells have an intrinsic ability to migrate around more. And this was actually indeed very true in the systemic sense, because if you flip the mouse over, a mouse that's been infected on the left side, flip it over and then look on the right side, for the presence of the memory T cells, you see CD4 memory T cells in these never infected distal regions of skin. They're moving around, they're in the dermis, and they're moving around quite quickly. And then in this situation, you hardly ever see any CD8 T cells, suggesting that there's a really quite pronounced difference in the ability of the CD4 or CD8 T cells to survey these peripheral sites. And we tested this more specifically by taking memory CD4 or CD8 T cells, normalizing the numbers and transferring them into mice, and then see where they went. And we see a predominance of CD8 T cells going to the spleen. This is a ratio of CD4 to CD8. There's more CD8s localizing in the spleen, but there's considerably more CD4 T cells localizing to tissues such as the skin and the vagina, suggesting again that there's a preferential difference in the ability of CD4 versus CD8 memory T cells to go through the tissues. Now, this is also very interesting if you look at the molecules expressed by the CD4 T cells. They suggest that they're able to get into these different skin sites because of the molecules they expressed. It's really the effector memory CD4 T cells that express the skin homing molecules ESL and PSL, E-selectin ligand and P-selectin ligand. Whereas the effector memory CD8 T cells were largely low for these two populations, suggesting that they're not intrinsically more able to get into the skin. But what is interesting is the fact that the central memory CD8 T cells do apparently express a decent amount of these two skin homing molecules, suggesting that maybe upon reactivation, when these cells then lose the expression of CD62L, they might then be able to get into the skin very quickly upon re-stimulation. But they're not doing that in the steady state. And we know from work done by Laura in the lab that the ability of cells to get into the skin upon reinflammation, in this case using the hapten DNFB as an inflammatory stimulus. The mice were infected with HSV, so they had populations of GDT and GBT memory T cells in the circulation, and then 30 days later stimulate a distal skin site, one that won't have resident memory T cells present, with the DNFB. And you find that although there's considerably more GBT CD8 T cells in the circulation and in the spleen, the cells that first come into the inflamed site are predominantly CD4 T cells, suggesting that their ability to migrate around allows them to get into that site more quickly. And blocking EMP-selectin can reduce that early entry of the CD4 T cells into the skin. And what's interesting is that this differential localisation and differential migration of the two memory T cell subsets is really not just skin-related, because if we infect mice intravaginally with HSV and then look at the memory T cell populations that are formed, we find both CD4 and CD8 T cell memory cells in the vagina, but the predominance of CD8 T cells is in the epithelial layer of the vagina, and the CD4 T cells are really predominant within the lamina propria, very much like the dermal-epidermal separation that we see in the skin. Not quite as black and white, but still very, very suggestive of the fact that the differential migration and the differential localisation of these two subsets is really holding true in different tissues. And what I also find very interesting is that if you treat mice with the contact sensitizer DNFB, so you don't need to use a virus or anything like that, treat them and then you get priming of CD4 and CD8 T cells in this case because it's a hapten. And then look at the memory phase, you find lots of CD4 T cells in the dermis, and you find CD8 T cells residing in the epidermis, suggesting that this differential localisation really holds true for a number of different situations. And what I also find quite interesting is if you just take a mouse, not immunised in any way, obviously if our mouse house is like anyone else's, it's not exactly a sterile mouse, but if you just look for CD4 T cells, you find them predominantly in the dermis. You don't really find CD8 T cells in the skin of unimmunosed mice. And similarly in the vagina, you find plenty of CD4 T cells in the lamina propria and the occasional CD8 T cell in the epithelial layer, suggesting that endogenous T cells also, in responding to antigens in the environment, can also undergo this different pattern of migration. And then lastly, knowing what we know about the ability of the CD4 memory T cells to migrate around the body, and then looking to see whether or not that's protective, using antibody-deficient mice infected with HSV, and then depleting either the CD4 or the CD8 T cells prior to secondary infection of the mice, we can show that the depletion of CD8s really has no significant effect on the ability of these animals to be protected from reinfection. But if you deplete the CD4 T cells, they're no longer protected from reinfection, suggesting that at least the early phase of a secondary response is really predominated by CD4 T cells if there aren't resident memory T cells initially in that skin site. But if there are, the TRM do mediate protection, and this is the original work done by Thomas, showing that the presence of TRM really allows the mice to be protected, and the absence of antibody or CD4 T cells, suggesting that TRM can really do a lot if they're localised in that site. And so what we started to look at was how TRM actually behave when they see antigen and what they're actually doing to protect an animal from reinfection. And what we know from very initial work is that that TRM can respond in situ to antigen, and they do so very specifically, i.e. if we put on the GB peptide, this is dissolved in cream and just put on the skin, the TRM quickly start to round up, and some of them start to get into the dermis and start moving around very quickly, so they're responding quite dynamically to the presence of antigen. But they don't show any change in movement or morphology if we put on an irrelevant peptide, such as from ova. And so we know that the TRM can do quite a lot very quickly if there's a reinfection. As early as 2 days after infection, they start to reduce the amount of virus in that site by up to a log in a very short period of time. And what we're starting to do is actually image the resident memory CD8 T cells as they reactivate to the virus. This here is using a GFP tagged HSV, and you can see here this ring here is the keratinocytes around a hair follicle that are infected by the virus. And these long, thin structures are the nerve fibers that are infected. As the virus is emerging from the fibers, it most likely starts there and then infects the keratinocytes. And you can see that the resident memory T cells have started to do quite interesting behavioral differences from when they were initially just localized to the epidermis. They're now penetrating into the dermis, and they're moving around and they're interacting quite closely with the virus-infected cells, presumably killing them and clearing the virus. And this is before the appearance of cells coming in from the circulation. And then a little bit later, you start to see massive numbers of CD8 T cells appearing, and they're killing virus-infected cells. And what we're really trying to determine now is what the general relationship is between the resident memory T cells in this response and those that are drawn in from the circulation. And so the model that we've been working on, the working model that we published, suggests that in a site such as the skin where you have an infection, after the infection is cleared, you get resident memory T cell, CD8 T cells, forming in the epidermis or epithelial layers, whereas CD4 memory T cells don't localize to the epidermis, they localize to the dermis. They move around much faster, and because of their position in the dermis, they're essentially in contact with the circulation, or they can enter the lymphatics, continue to circulate around the body, and probably come back into the skin as they go in their migration pattern. That allows them to access the lymphoid tissues, probably other non-lymphoid tissues as well, where of course you also do have populations of effector and central memory CD8 T cells, just not the resident memory T cells. And this of course allows the CD4s to also get into distal skin sites as opposed to the resident cells which just remain at the site of infection. And we also know when we're starting to examine how the TRM actually behave and how they form, that their morphology is quite unique from other cells in the skin, and they of course remain there for long periods. They show a really sustained surveillance or patrolling of the epidermis, which probably allows them to actively seek out any reemergence of the infection. And their preferential localization at the site of prior infection, at least to me, suggests there's probably a local niche allowing them to stay there and allowing them to survive. Something like IL-7, for instance, might be a good candidate. And they're independent for the circulating memory pool, but they can respond locally to antigen, and we know that they clearly contribute to protection from reinfection. It's just a matter of now quantitating how much they contribute. So that just leaves me to thank everyone involved in this, in particular Bill Heath and Frank Carboni, Thomas Gebhard, who started all the TRM stuff, Eliza, Laura, Liv, Azad and Nick, as well as Andrew Brooks and Paul Whitney for the GDT2 mice, all of the lab members of the Heath Carboni lab as well. And funding for this for me was from the ARC and from others from the NHMRC. So, thank you.
Welcome again. Oh, I'm going to say, you attribute a lot of differences to CD4 CD8 responses on the basis of the transgenic, but of course they will have different sensitivities to the antigen. There could be an affinity difference that might affect their phase. And you did show the Hatman experiment with residual cells in the non-sterile mice. But I wondered if you could follow, just by sections, the response to HSV of the host T cells in a non-transgenic system and show the same pattern in these sections between the CD4s and CD8s.
Do you mean in a specific fashion, tetramerically, or just total?
Just total.
We can, and we sort of have, in a very crude fashion, and we we see a similar thing. You see CD8s now appearing in the epidermis, and you don't really see CD4s at memory. And you always see a predominance of CD4s in the dermis. So I guess that sort of answers that, but we haven't really done it very well. Yeah.
I just clarify something. All of the cells, including those memory CD8s in the epidermis, seem to have a similar form, dendritic form and slower movement and so on. Do you interpret that as being dictated by that very compact, half-dry, low-ceiling environment, or is it an adaptation to that environment? In other words, if you took them out of that environment, would they maintain that form, or would they behave like other T cells in that case? Have you got any feel for which way it is?
I would say it's probably a bit of both, but if you take them out of that environment, they tend to die, which is a difficult thing to study in many ways. We haven't really done it and then short-term looked at whether or not they revert to a different morphology and a different movement pattern, but my guess is they would, which would suggest it's really just the environment they're in. But that said, the gene expression pattern of these cells, which is something that Azad is doing, is very different from that of a circulating memory T cell. So there's a lot of changes that obviously occur within the T cell once it gets into that site.
Do you know how long after infection the resident T cells have you done any long-term experiments to see how long they stay in the skin after infection?
Well, I've done well over 300 days and they're there, and I think Thomas has probably done even more. As long as a mouse lives, it's probably a lifetime of a mouse.
How long, Thomas?
2.5 years.
So, and that's pushing it for a mouse, right? So, very long term. Yeah.
If you do Laura's experiment, right, where you infect with HSV and then you put in TNFB to get your effector T cells there, and you compare that to a mouse that you don't put TNFB on, and then reinfect with HSV, is the recruiting faster where you have resident memory T cells?
I think she's tried to do that, and it wasn't clear. The suggestion would be, yeah, you would think it would be faster, but it's hard to really quantitate that well enough in the short period that they're being brought in. So we don't really know the answer to that. We have these Katie mice which will allow us to shine UV light on the cells that are in the skin, and then we can track who is actually coming in. And then we have some idea of the recruitment versus the resident, i.e. those that were already there versus those that are coming in. And that'll give us a bit of an idea of what role that plays. But in terms of whether or not they come in faster in that case, we don't have really good data to suggest that.
Scott, the paper that you recommended for the student discussion group showed, I believe, using a vaccinia and ova model and OT1s, that in fact the CD8 positive TRMs were accumulating at distant sites in skin, but they either didn't look or didn't find CD4s. What do you think the difference is there?
Well, yeah, they didn't look at CD4s, and of course they're using a different virus, but they also didn't really look to see if they were actually TRM versus TEM. And in many of the cases, especially the latter experiments, the massive increase in numbers they see in different skin sites could well be just TEM rather than actual TRM. And people like Dave Massapus have done these multiple priming experiments, and you get massive increase in circulating TEM numbers, which presumably are in the blood vessels all throughout the body. So if you have so many TEM circulating around, they can actually add quite a lot to the numbers that they were getting. So my assumption is that they just weren't quantitating well enough to say that these are TRM in the distal skin sites.
On that note, you said that most of the virus is cleared after 7 days, but have you used your GFP strain of virus to actually see that these are
Well, we certainly can't detect it by PCR or by plaque assay. We haven't done it with the GFP virus. You mean look at memory, for instance? Or we haven't done that. My assumption would be that we wouldn't see anything because usually PCR is more sensitive. Yeah.
Any more questions? Can I make a plug, by the way, for all PhD students? One, please ask a question. Two, please attend Matt's discussion groups. I think that would be, well, once I've attended at least, I think the students got a lot out of it. So please attend Matt's discussion. Well, I guess it's been a real treat, Scott. Lovely photos. Thank you.
Thank you.